Hi friends, today we'll be talking about enzymology, metabolism, the vitamins, the cofactors. Yes, so today we'll be talking about one of the common cofactors for various dehydrogenases, and that is your FAD or your FMN, that is your flavin adenine dinucleotide and flavin mononucleotide. So let us talk about these vitamin component of this particular cofactor. So the vitamin which we will be discussing today is your riboflavin. Riboflavin, that is your vitamin B2. Now this particular vitamin riboflavin, as you can see here, it comprises of this a peculiar ring system which is referred to as the isoalloxazine ring system. This particular vitamin, you will find it gives its intense yellow color, and this particular intense yellow color of this particular vitamin is because of this isoalloxazine ring system. You must have seen the yellowish coloration of milk, of the egg yolk. That is because of this particular vitamin, that is your riboflavin. So this particular riboflavin, as you can see, so the vitamin which we are discussing today is your riboflavin. So riboflavin or your vitamin B2 containing this isoalloxazine ring system and this particular chain. Then you have a particular cofactor form. Every vitamin when it what you call as functions as the transient intermediate carrier of any particular group and is associated with the enzyme, it is first converted into a particular cofactor. So riboflavin, it acts as the cofactor, or the cofactor form of this particular riboflavin is your FMN or your FAD, that is your flavin mononucleotide or flavin adenine dinucleotide. Both of these, they occur in two forms. One is the oxidized form, another is the reduced form. So you have FMN and FMNH2, or you have FAD and FADH2. The group that is transferred, most of these what you call as vitamin B complex groups, when they function as the cofactors, they serve as a transient intermediate carriers of particular group. In this particular case, this riboflavin, when it acts as a cofactor in the form of FAD, it acts as a transient intermediate carrier of hydrogen atoms. Remember, so the group that is transferred is your hydrogen atoms. The enzyme, now there are many enzymes, various oxidases, various dehydrogenases, they use these particular FAD as a tightly bound prosthetic group. So this particular FAD will be tightly bound to the surface of the enzyme with the help of covalent bonds. So it acts as the prosthetic group of various dehydrogenases. To give a specific example, you have the enzyme succinate dehydrogenase. So succinate dehydrogenase is an enzyme which converts your succinate into fumarate. So there, when the hydrogens are removed from the succinate to introduce a double bond to form fumarate, the hydrogen atoms are transiently carried by this FAD. So you have the vitamin riboflavin B2, its cofactor form FMN or FAD. The group transferred is the hydrogen atom. An example of an enzyme having this particular vitamin as the cofactor is your succinate dehydrogenase. If you look at this structure, you will find this is the structure of FMN, that is your flavin mononucleotide. You can see this is a pentose which is then attached to this particular phosphate, which gives it the structure of flavin mononucleotide. So here you can see this is your riboflavin, right? And then this is your FMN. FAD, a bit complicated structure, but then you will find that it comprises of this isoalloxazine ring system. It comprises of this ribose phosphate. It comprises of this particular adenine. So when you have two what you call as nucleotide attached, so a ribose sugar to one nucleotide, another ribose sugar to this similar kind of a nucleotide nitrogen containing ring structure, that is riboflavin. Therefore it is referred to as flavin adenine dinucleotide. So you have this ribose attached to this ribose attached to this, and a phosphate in between, so a dinucleotide. So it is a flavin adenine dinucleotide. Though this looks a very what you call as a complex structure here, but we are going to focus on the active ring structure here, the one which is going to act as a transient carrier of the hydrogen atom is these two what you call as your nitrogens. So when you have a particular substrate which is reduced, say for example, you have a particular substrate which contains what you call as hydrogen atoms, it is in the reduced form. So you may have, if you look at this, yes, you may have a S which is containing your 2 hydrogen atoms, then this may combine with your FAD, this substrate which is reduced. So this represents the reduced form, this represents your enzyme containing FAD. This reduced substrate is oxidized. So now this particular reduced substrate is oxidized, and your FAD which is there, that is converted to your FADH2. So hydrogen atoms, they are transferred from this particular substrate to this particular FAD. So your FAD gets converted to FADH2. So where exactly this particular hydrogen goes? Now if you look here, to make it more simpler, let us what you call as look only at the ring structure where the actual reaction takes place. Yes. So here if you look at this particular ring, let us consider this. Let me represent the entire ribose component with R, and say let us this is attached to the enzyme. You have a particular substrate now which is having hydrogens. So this is your reduced substrate. So reduced substrate combines with your EFAD component. This where it will be oxidized. So your substrate, it will be oxidized. So this will give a oxidized substrate. The hydrogen atoms, they are transferred to this particular ring here. So you'll have a change in a structure, a slight bit change where the hydrogens, they will be attached. So you'll have hydrogens attached here, and there will be an introduction of the double bond here. So actually you are going to focus here now. So this is where your hydrogens, they are attached now. So this is a transient conversion of your FAD. So this represents your FAD, and this represents your FADH2. So it is these nitrogens which are actually going to act as the transient intermediate carrier of your hydrogen atoms. So hydrogen atoms from your substrate, one attached here, one attached here. The change in this particular structure, the double bond is introduced here, these double bonds are removed, and there is formation of this particular FADH2. The specific example which we told, if you look at that, your succinate is converted to what is called as your fumarate. So your succinate, it is being converted to your fumarate. So if you look at that, you will find, if you look at the structure of a typical succinate now, so a typical succinate, we know it is, yes, so this represents your succinate. So succinate, right, it combines with a FAD, enzyme bound FAD. I told that FAD is tightly bound to that particular enzyme as the prosthetic group. There will be an introduction of the double bond here. Hydrogen atoms, they will be removed, and your succinate, it is converted to your fumarate. And this particular FAD will be converted to your FADH2. So your succinate is oxidized to form what is called as fumarate in the presence of enzyme succinate dehydrogenase. So succinate dehydrogenase, it converts your succinate into fumarate by introduction of a double bond here and removal of hydrogen atoms which are accepted by FAD to form FADH2. This FADH2 can later on give the electrons to electron carrier proteins in your ETC, which will be again oxidized back to FAD, which can be used further. So vitamin, riboflavin, the cofactor, FMN or FAD, the group transferred, hydrogen atoms, and the enzyme succinate dehydrogenase. Stay tuned with me, Professor Girish Kukreja, for more in microbiology, enzymology and metabolism. Thank you.